EMSA. You might ask, why do you have sperm DNA in the cold room? It's for the EMSA, or gel shift assay. So basically, the gel shift assay is a way if we can tell if a protein is binding to a specific piece of DNA. And so you might be used to agarose gels for running DNA and separating them by size, and SDS page gels for running proteins and separating them by size. Well, with the EMSA, the gel shift, you're typically running a page gel because it gives you better separation. But if you have like a huge DNA, then you can use an agarose gel. But the idea is you don't want to add the SDS, so you're not unfolding the proteins. What you're doing is you're trying to see if the protein is going to interact with the DNA. And so the basis with the gel is that the gel is going to separate things by size. Well, a DNA by itself is going to be a lot smaller than a DNA plus a protein. And so when you run a DNA alone, it'll run at one size. Then if you add a protein, boof, it'll be run at a higher, it'll run like it was a bigger thing. And if you label the DNA and then detect where the DNA is, you'll see a low band when the DNA is not bound and a high band when the DNA is bound. And so we say that it is shifted, a gel shift. So that's the basic idea, but of course there's a lot more details, and there are controls that you need to do to make sure that this binding is actually specific between the protein and the DNA. And then there are also things that you can do to make sure that that protein is really what you think it is. And so I don't keep you waiting too long, basically the salmon sperm is just a really good source of a lot of DNA. And this DNA has a bunch of repetitive sequences, and it's really not specific and stuff, and so basically it's like sheared up, so it's like little pieces of DNA. And then you could use it as a non-specific competitor, so that if your protein just like binds any old DNA, it's going to bind to this, and this isn't labeled, so it's not going to show up. So it's going to look like your protein isn't binding to the specific DNA because it's going to be binding to all this non-specific DNA that you've like sopped it up with. But if the protein binds to DNA specifically, so it likes that piece of DNA, that labeled piece of DNA, then it doesn't matter if you put in this competitor, because it's like, no, I don't like that competitor. I like this specific DNA, so I'm going to bind this specific DNA. And when it binds that specific DNA, since that specific DNA is what you have labeled, now you're going to see the shift. But if it was just binding any old DNA, now it's going to be binding to the sperm DNA, and so you won't see the shift. So most of the time in my posts, I've been talking about proteins and nucleic acids. So most of the time in my videos, I've been talking about proteins and nucleic acids. So proteins, they're made up of amino acids that fold into these chains. Nucleic acids, they're made up of, these are DNA and RNA. They're made up of chains of nucleotides, and they hold like genetic information and stuff. And then the proteins are the thing that actually like do stuff a lot of times. Gross oversimplification. But actually, like proteins and DNA and proteins and RNA are hanging out a ton. And so often we want to figure out who's hanging out with who, and like how tight their interactions are and that sort of thing. And a way that we can do this is with the EMSA, or gel shift assay. And so EMSA stands for Electrophoretic Mobility Shift Assay. And so electrophoresis, we're used to electrophoresis. We've talked about it all the time, if you've been following along. So we've talked about agarose gel electrophoresis, where we have these slabs of this gel made of this sugar agarose, or agrobios. And then we've talked about PAGE-polyacrylamide gel electrophoresis. And we've used this to separate proteins. We use SDS page to separate proteins. We've used urea page to separate RNA. And the basic idea with both of these is that you're using this electric gradient to get molecules to move through a gel mesh. And so when they're moving through this mesh, they're going to get tangled up in the mesh, and then that'll make them travel slower. So the bigger things are going to travel slower than the smaller things. So the smaller things are going to end up down here when you turn off the power, and the bigger things are going to be stuck up here when you turn off the power. And so in order for those to work how we normally work them, so where we can have like a molecular weight standard, so we have this thing of known sizes, and then we can line up, we can run that alongside our things and look and see where we would expect things to be. Well, that expects your things to all be linear, so not all folded up, which is
a big thing for the proteins, and they also expect your things to be uniformly negatively charged, which is cool for DNA. And so you need that negative charge because you're going to be using this positive charge to entice it to move through the gel. And so your DNA already has this negative charge in its backbone. Proteins can have various charges though, so they might not go if you don't have the SDS, this detergent, to give them a negative charge. But the real thing about the really important thing about this detergent is that it's going to unfold or help denature the protein. So it's going to unfold the protein. But if you're trying to study how a protein is interacting with the nucleic acid, so with like DNA or RNA, well, the protein is going to be interacting in this folded state normally, right? And so you don't want to just like try to get them to interact with an unfolded protein. So instead of adding SDS, what we're going to do is we're going to run a denaturing page, so like a native page. So we talked about native page the other day, but basically here you're not unfolding the protein, you're running it as is. And this will like keep protein complexes together, but it also keep protein DNA and protein RNA complexes together. And so we talked about how you can see like a shift when you had proteins bound together. When there's a complex, they'll run as a higher, like they were a higher size. Similarly, when you run an EMSA, so when you have a DNA bound or an RNA bound, it'll run like it was a bigger size. So when we're running these gels, so we're going to be running a page gel typically. So often we're doing like a TBE page. So tris or something, I don't know. But anyway, so it's not an SDS page gel. We're not using detergent. We're not denaturing the protein, but we still want to use a page gel because it's going to give us better separation. So when we use like an agarose gel, we're typically using like big fragments of DNA, and we don't need like super duper clean resolution and stuff. So like we don't care if the pores are like a little varied in sizes or whatever. So the page gel, you get a more uniform pore size and you get smaller pore sizes. So you get like a tighter mesh, and this is going to help you separate things. And so often what we're doing is these probes, so the DNA or RNA we're using, they're going to be small pieces, not like huge pieces. So they can be big pieces. And so often if you have like really big pieces, you can use like an agarose gel. But typically you're doing like a page gel with like smaller pieces. And the important thing is that you need to label the pieces. And so these things are going to be acting as your probes. So you can label this in different ways. So you can label it with like the classic way is with P32. So with radioactivity. And so this is the method that like we use here. It's super duper sensitive. So that's a really great thing about radioactivity is that it's really, really sensitive. And it doesn't change the, like, you're not adding a big bulky thing onto the end. It acts just like it was normal DNA or RNA. It just has this radioactive isotope of phosphorus. So it has like a different number of neutrons. So it'll give off this radioactive decay. But your like the proteins and all like don't really realize that anything's up. But so a lot of people want to avoid radioactivity, and so sometimes you use like fluorescence. So you can stick a fluorophore. So basically it'll absorb light of one wavelength and then give light off with another wavelength. So you can stick in you can like shine light at it at the wavelength that it likes, and then measure and see if it's giving off the wavelength that you expect to give off. And so that's the idea with when you fluorescently label something. And then there are also methods of labeling where you can label it with something that then you can like add something and then that'll bind to the something that it was labeled with, like biotin or whatever. So there are different methods. But the reason why, so you might be wondering like, well, why don't you just use like one of those like why don't you just use like ethidium bromide or one of the like fluorescent stains that you normally use with your agarose gel? Well, we want something that's really, really sensitive. And so these, like with especially radioactivity, with fluorescence, you're going to have a lot higher sensitivity. And with especially since you're often having like shorter pieces. So the way like ethidium bromide and those dyes work is they're going to be length dependent. And because they're going to be binding like along the length as opposed to just like at an end. So this means if you have a shorter piece, it's going to have less dye bound. and it's going to be like, so you're going to get a weak signal. We want to get a strong signal, and we want to be able to detect really small amounts, because we're going to be trying to see where that how much of the band is shifted versus unshifted. And so we want something that's going to give us really accurate data and really sensitive. And so that's why we labeled the probes. So we're running a page gel, but we're not looking for proteins. We're looking for the DNA or RNA. So it can get kind of confusing if you're not used to running page gels for DNA or RNA. But that's what we're going to be looking for. But we're also having proteins in the mix. And so we're going to infer the binding of a protein based on a shift in the gel. So basically, if the basic idea is that if you don't have your protein, then the DNA is not going to, it's going to run at a specific size. It's going to run like it was really small. And so you'll see it far down in the gel. If you add the protein and it binds to that DNA, now you're going to get a shift. And so you'll see a higher band, because remember, you're looking at the DNA, you're looking for the DNA. You don't see that the protein's here or not, you just see the band, the DNA band move. And so like, it didn't just like magically move, it moved because it bound to something. And so that something is the protein. And so here we're talking about if you have like a super pure, we'll get into the details later, but if you have like, you're starting with just like really pure protein, really pure probe, then that's the only thing that could be making it shift, right? So it's the protein, it's binding your protein. And if they don't bind, then the protein, you're not going to see the protein. And so you're just going to see the DNA where it is as if it were running alone. So there's actually tons of copies of all of these, and the strength of the signal is going to depend on the number of the probe molecules you have. And then, but the important thing is the position of the signal is going to depend on whether it's binding the protein and how many copies of it you're binding. So because this is a native page, because this is denaturing, because your proteins are folded up, because proteins have different sizes, have different like charges. And so now charge is going to come into play because we don't have that SDS negatively charging things. And then if you add the DNA, which is negatively charged, so now your protein is going to be bound to this negatively charged thing, which thankfully will get it to run through the gel, even if it wasn't negatively charged to begin with. But basically, you can't really predict easily where this protein is going to end up. And so you can't just use like a molecular weight standard to be like, oh, is that really my protein? And so we'll get into how you can use like antibodies to induce a super shift. So basically, you use an antibody that will bind to the protein specifically, and this will cause a shift, and that'll let you ID your protein, which can come in really handy, especially if you don't start with a pure protein mixture. So if you start with like a lysate or something, so just like you want to see like what in the cells it's binding to or whatever. So you can do like this and then do like a Western if you have suspicions about what it might be. Or you can use this, add an antibody and see if you see a further shift, which we call a super shift. But so with the EMSA, like, so how far it'll bind is also going to depend on how many copies of the protein. So just like it'll bind, it'll shift further when it binds to the antibody, which is a little protein anyway. When it binds, if it binds to like another copy of a protein, it'll also give you a super shift. So you'll get a higher shift than if you just had a single protein. But you can't tell if it's going to be, if it's like two binding sites. So if it's two proteins bound to different sites on the probe, or if it's a single or if it's two proteins bound to each other, but bound to a single binding site. So if you want to find out that you can like mutate the probe, and so you can try mutating the probe. And so if you do that and the stoichiometry changes, so the stoichiometry is like the ratio of the probe to the binding protein. So if you make a change to this probe, you change the sequence, and then the stoichiometry changes, then you have two protein probe interactions. Basically, if you disrupt one of them but not the other, then the other could still bind, but not this. But if it doesn't change, you could have either. You could be that you have this protein-protein interaction, or that you just didn't mutate the right spot. But you can introduce mutations in either the probe or the protein to try to figure out where they bind within the specific sequence. And so like old school ways or whatever, like you take like huge pieces, you like take huge pieces of DNA, like before all this sequencing and all of this. And then you would try to figure, you take these big chunks and then see, okay, with the protein in here, would it bind somewhere in here? And then you can cut that chunk down into smaller and smaller chunks. But basically the smaller the piece, the less possible sites there are for binding. So let's look at what you do. So how you can get information, use the EMSA to get information about how strong the affinity is, so how much they like to bind to one another. And so basically, as I talked about in past posts, like anything will bind anything if you give it enough, if you like stuff enough of it in there, like, because if you have like a ton of stuff, like every time you you run into it, even if you don't really stick to it very well, and then it unsticks, then there's another copy to stick. And so it's going to be like you're always bound. But if you really like that thing, then it's not going to take as much to bind to it all. Or in the case of when we talk about affinity, we're usually talking about the amount to bind half. So this KD, this dissociation constant, the concentration of the protein that's required for half of the probe to be bound. And so in order to find this value, we need to test a range of concentrations of the protein. And then the smaller the value of the KD, the stronger the affinity, because you didn't have to add as much to get to that halfway point. Because when these proteins is binding to the DNA, it's going to be binding stronger. And so it's not like it's just like constantly letting go, and then you need to have another guy come in and like run into it. So the KD is going to depend on both like the on rate, so like how fast it binds, and the off rate, how fast it lets go, and as well as the concentration and stuff. And so I'm not going to go into it here, but basically know that the KD is going to be this value at which half of the probe is bound in these assays. And so more generally, it's just like half of one of the binding partners. The basic idea is that if you have no protein, all of your DNA is going to be unshifted. If you have a ton of your protein, all the DNA is going to be shifted. And so remember, we're looking at this is what you actually have in there, this is what you see. And so you're going to, I mean, like not in terms of the actual like height or whatever, just the quantity of stuff. But basically, you are going to see it all shifted if you have a bunch of protein. And then you do this gradient where you're trying to find the point at which half of it is bound, and this is going to tell you the affinity. And so you'll see that the band at the bottom, so corresponding to the unshifted DNA, that's going to decrease in its like strength and its intensity. It's not going to decrease in like you're not going to, when you say shift, it's not like you gradually see a shift upward unless it's binding like one protein and then it's binding two proteins. You won't see like the shift thing. You won't see like the shift in progress type of thing. Instead, it's kind of like more like one or the other. And so, but one is at the expense of the other because you have a constant amount of this, the labeled stuff. And so you're going to have a decrease in the unshifted as you have an increase in the shifted. And then that is like halfway point. This isn't exact, but it would be somewhere like in the middle, where you have these like equal bands. And then this would be your KD. Yeah, sorry, this isn't like totally accurately drawn, but I was tired and working. But anyway, so if you have something with a high affinity, it'll take less to get to that halfway point. And so you're going to get a leftward shift. So basically you'll get more at the top. This will disappear faster, and this will increase faster if you have a high affinity. But if you have a low affinity, then it'll require more protein, so you'll get this rightward shift where it's going to take more for the same amount to be bound. And so you typically like optimize your protein concentration range to get a nice curve where you can clearly see where there's like none bound and all bound. And then you can use like fitting software to get the KD from that. Important caveat is that the EMSA, so it's not like a true equilibrium binding assay. And so the KD is like the apparent KD or the approximate KD. So when you're mixing these things together, the KD is like this thermodynamic constant or whatever. And so it think it's relying on the system being at equilibrium so that it's kind of like found its happy place. When you're running these gels, the things like your sample is running through a gel. And so it's kind of getting diluted. So thankfully there's this thing called the caging effect where it's like stuck in the gel. So even if it like comes apart, like this high local concentration, even though it's getting diluted through the gel, there's this high local concentration because you have it like stuck in this, like in the gel, like it can't go far from one another. So this will help you, but it's still not giving you like a true equilibrium value. Plus you have like your sample preparation and then you're putting in the gel and stuff. And so there's some variables. And so it's not like super duper accurate, but it can give you an idea. So you are going to need to run some controls because you want to make sure that that binding is specific. And so basically it could be that that protein will just bind to any old DNA and that or RNA. And that wouldn't really tell you, I mean, it would tell you that it could bind nucleic acids, but it wouldn't tell you if it's specifically binding to that specific nucleic acid. And so typically what we do is you add competitors. So if you want to test for like specificity, you can add an unlabeled non-specific competitor. So remember that we're only going to be seeing the labeled stuff, which is another reason why it's helpful to have these labeled probes as opposed to just staining for all nucleic acids. So we have this unlabeled non-specific competitor. So this can be like random DNA, like that salmon sperm I was talking about, or like a synthetic alternative like poly-DIDC, or a mutated version of the specific probe sequence. So if your protein binds to DNA or RNA non-specifically, then it's going to bind to this generic stuff because you're adding a big excess of it. And so it's more likely to run into this. And so if it likes it, or if it just binds non-specifically to anything, it's going to bind to this. So this is going to basically use up all of your protein. And so then when you add your labeled stuff, it's not going to bind that. And so you want to add the competitors before you add your labeled stuff. And so you don't accidentally just like have your stuff, if it was like holding on tightly, but it would still bind to other stuff, then it could influence how much of you would still see a bunch of the labeled stuff bound. But if you do add the non-specific stuff first, then you are going to get the protein like used up, and then you won't see this shift. But if your protein is binding specifically to that other stuff and it doesn't care about this non-specific stuff you're throwing in there, then you won't see a change in your band and you'll still see it there. So if you have specific binding and you put it in a non-specific competitor, you're still going to see that same shift. And okay, so like caveat, like, so if it's like you have a ton of like way too much of the non-specific stuff, you still might see like a decrease in the amount shifted, just because of that thing we talked about, or if you throw a ton of anything, it'll like, some of it'll just be bound to bind because you just have so much of it. And so you have to kind of like optimize the concentration. What if you were to add an unlabeled specific competitor? So here we're talking about, this can be something that the protein does like to bind. So something you either know it likes to bind. So if there's like a known binding sequence for this thing, or it could be the same as your labeled thing, but without the label. And this, you do the same type of thing. But here, if your protein, even if your protein specifically binds to that thing, now it's going to bind to the specific thing that's unlabeled. And so it will look like you're going to lose the band corresponding to the shift from the binding. Okay, so that's going to tell you about this. And then you can also test like try competing with like different sequences and that sort of thing to get better idea. And you can introduce mutations like we had talked about into the, introduce mutations into either the probe or the probe to try to figure out where in this probe it's actually binding to. So that'll tell you about like the binding location and the binding specificity and stuff. But what about the protein? So when we're doing this, we're only looking for the probe we're not looking at like at the protein. We're just inferring that, we're just like assuming that the protein is causing it to shift, right? Because something's causing it to shift. It's not just like growing up. And so we're inferring that it's this protein that we think it is. If you start with a really, really pure mixture, if you start with a really, really pure protein, then this is less of an issue. But if you start with like a less pure protein or with like a cellular lysate, so basically cellular insides, then this can be a really big issue, because those probes the signal is so sensitive, and you're only going to see the probe, not the location of the not the location of the proteins or the and because we're running like a native page, we can't really easily predict where a different protein will run. And so it's really important that we can then identify what protein it actually is. And if we know what protein we suspect it is, then we can do this with antibodies. It's a little bit of a contaminating protein that really likes to bind the probe, then you're going to see like a band for that. And remember, this is a native page, it's denaturing, so we can't really predict very well where a protein is going to run when it's bound to this thing. And so we don't know if that band is actually for the protein that we think it is. And so there are ways that you can control for this. And so one of these is doing adding an antibody to induce like a super shift. So basically, if the antibody binds, then you are going to it's going to have this bigger complex, and so it's going to travel even slower, and you're going to get a higher up band, this what we call a super shift. There are other ways, such as doing like a Western blot afterwards. So basically you can do, actually, there's like a combined like Western, Northern, or Southern, I don't know. But anyway, basically you transfer the nucleic acids to a membrane, and then you scan that and see where the nucleic acids were, and then you transfer the proteins to a thing, and then you like probe with antibodies to see what the protein is. So typically, this is also really useful if you're starting with like a crude lysate or something, so just like the cellular insides. And so you think you know what protein is binding, so you have a strong suspicion, but there's a bunch of other proteins in there too, so you might actually see like multiple bands if you have multiple different proteins binding. But if you have a band and you think it's we're trying to find like, does this one actually correspond to the protein that I think it does, then the antibody can tell you that. Also, if you know like where on the protein is responsible for binding and you mutate that protein, then you won't see that shift. And so it all depends on like your system and what you're doing, and whether you're working with purified proteins or lysate or whatever. And so, yeah, so these have been used a ton in the past, a lot more with the like cellular fraction. So you could see like take cells like different time points and see what's bound to a specific piece of DNA and stuff. And so there's all sorts of cool things that have been done with the gel shift. And this is, but you want to make sure that you do your controls to make sure that your binding is actually really specific, and that it's the protein that you think it is. So I've talked before about how I use this method called a slot blot, where you basically mix a protein and a labeled DNA or RNA together, and then you mix different amounts and you separate them with this vacuum filter apparatus, where there's like a membrane on top, a nitrocellulose membrane to capture the protein, and then below it is a nylon membrane that is going to capture the free DNA or RNA as it flows through. And then you vary the amounts of the protein to try to find the amount at which half of the probe is bound, which is a value we call the KD, which tells you about the affinity. So with an EMSA, it's a different way to figure out the affinity. But you're still going to use a concentration gradient of your protein and keep a constant small, low level of the probe. So typically to get this concentration, like gradients or whatever, you do a serial dilution. So you like half, that sort of thing. What does the EMSA have? So one of the things that the EMSA has there are certainly this. So the slot plot is typically more like accurate in terms of determining the actual like constants and stuff, but it's even that's not as good as like true equilibrium methods and like that sort of thing. But anyway, I'm not going to get into that. But one thing that the EMSA can do that the slot blot can't is that if you don't, for the slot blot, you need to have like a really pure protein, and a really you need to have a really pure protein, because if you, this basic idea of how it works is this top, the membrane is going to bind any protein. And so you could, this, if your stuff could be, any protein could be binding and you would get the signal. So you need to make sure that you only have your protein binding in there in order to make sure that you are only getting you need to make sure that you only have your protein in there so that the signal is corresponding to your protein. If you don't have a super pure protein, then this is not going to work. But the EMSA would still work, especially if you combine it with like a super shift, so you can identify the identity of the protein. Because remember, you're looking only for the position of the probe. So you could have a ton of other proteins in here and it wouldn't matter as long as they didn't bind the probe. And that's another reason why to add like non-specific competitor is so that you can, like, any of those other like random proteins you have in there, if they like to bind DNA non-specifically, then they'll bind to this other stuff and not bind to your specific stuff and won't interfere. Another advantage of the EMSA is that you get that, you can get that stoichiometry. So when you do the slot blot, you don't know if the you don't know like how many copies of the protein are binding or whatever. So you don't get any idea about the stoichiometry, because it's all just kind of like protein. This would all just be this. All the stuff is just kind of like condensed onto a membrane, like flop it on there, right? And so you're not going to be able to tell if you have like one copy or two copies. You're not going to get a difference in the shift, but you can see that with the EMSA.